In the last video, we talked about the 48 nuclear receptors and how they regulate a specific set of genes, how each and every nuclear receptor is activating a specific gene. Today we will talk about one of those 48 nuclear receptors. That one is cortisol. So the cortisol has a receptor that is called glucocorticoid receptor. Cortisol is secreted by adrenal glands in response to ACTH activation. After activation of cortisol, the cortisol enter into the cell through the cell membrane and bind with its receptor, the glucocorticoid receptor. This glucocorticoid receptor is in inactive form, and when it binds with the cortisol, it is activated. In inactive state, it is bound with heat shock proteins. These heat shock proteins are specifically heat shock protein 90, which are actually molecular chaperones, and they helps in refolding of misfolded proteins. If there are no receptors in the cell for cortisol, then 11 beta hydroxysteroid dehydrogenase type 2 degrade the cortisol into cortisone, which is the inactive form, and cortisol is the active form of cortisol. In the active form, cortisol binds with the glucocorticoid receptor, and this receptor, after binding with cortisol, move from the cytoplasm into the nucleus through nuclear pores, and it has a specific binding site on the DNA, which is called glucocorticoid response element. This glucocorticoid response element is a sequence of the DNA to which the glucocorticoid receptor binds. The glucocorticoid receptor bind in the form of dimers. Like there are two glucocorticoid receptor will come into a cell to activate a specific set of genes in the DNA. So it activates the anti-inflammatory genes or proteins, both genomically through increase in expression of their mRNAs, and also through their activation in the cytoplasm. These anti-inflammatory proteins are then involved in the inhibition of inflammatory proteins in the cells. So we will just talk about how those inflammatory proteins are working. Cytokines like tumor necrosis factor alpha, IL-1, lipopolysaccharides, and viral proteins in the blood activates their receptors, and then those receptors activate the nuclear factor kappa B, which is actually inhibited by I inhibitory kappa B. This protein is inhibiting the active form of nuclear factor kappa B. So when these ligands or these cytokines activate the their receptors, they degrade the inhibitory kappa B and cause loss of the inhibition of nuclear factor kappa B, after which the nuclear factor kappa B activates their targeted genes, which are actually the inflammatory proteins. And the glucocorticoid receptor is actually involved in the inhibition of these inflammatory proteins expression as well, by a competitive binding with those binding sites where the nuclear factor kappa B proteins are actually binding. So in this way, it not only inhibit the expression of the inflammatory proteins, but also in their external activation is also inhibited by the anti-inflammatory proteins. So that's how cortisol is an anti-inflammatory protein. But cortisol along with glucagon is also involved in the gluconeogenesis. That is the formation of glucose from the proteins or amino acids. The glucocorticoid receptor binds with the phosphoenol pyruvate carboxykinase gene, and it upregulates the expression of this gene. And this protein is then involved in the conversion of oxaloacetic acid into phosphoenol pyruvate, and thus increase in glucose formation through gluconeogenesis and the release of glucose. So this is one way. The cortisol receptor is one of the 48 nuclear receptors involved in regulation of a specific set of genes. If you have any questions, you can ask me in the comment box and I will try to answer all of your questions. Thank you.